Numerous proteins, most notably the cytoskeletal protein actin and the actin-based motor protein myosin, must work in concert to power muscle contraction. Muscle cells contain numerous myofibrils, which consist of interlocking thick and thin filaments. The thick filaments are made up of myosin protein subunits, and the thin filaments contain actin, tropomyosin, and troponin proteins. Z-disc proteins serve as anchors for the thick and thin filaments. The protein titin functions as a flexible spring connecting myosin proteins to the Z-disc. According to the sliding filament model, thick and thin filaments slide past one another to shorten the distance between adjacent Z-discs during muscle contraction. In relaxed muscle, myosin binding sites on actin are blocked by tropomyosin and the troponin complex. Muscle contraction is triggered by the release of calcium ions from the sarcoplasmic reticulum. In the presence of calcium ions, troponin undergoes a conformational change that reveals the myosin binding sites on actin, allowing the myosin head domain, which is bound to ADP and inorganic phosphate, to bind tightly to the actin thin filament. When the myosin head releases inorganic phosphate, it undergoes a large conformational change called the power stroke that pulls the actin fiber along the myosin fiber. ADP is then released from the myosin head. After a new ATP molecule binds to the myosin head, myosin disengages from the actin filament. ATP hydrolysis returns the myosin head back to its starting conformation, allowing the cycle to continue. Muscle relaxation occurs when calcium levels in the cytosol decrease as a result of active transport of calcium ions back into the sarcoplasmic reticulum. When calcium ion levels decrease, the myosin binding sites on actin are blocked again by tropomyosin and troponin, returning the sarcomere to its original elongated state. The sliding filament model elegantly describes how force can be exerted at a molecular level to affect change at the level of cells and tissues.